All right, so in lecture 1, we looked at how eukaryotic cells came to be via endosymbiosis. Now let's look at how they evolved once they were eukaryotes. So they were single-celled to start with, but then they started to evolve further with a second wave of diversification where they got multicellularity. This gave rise to algae, plants, fungi, and animals. This multicellularity can be in many forms: colonies, collections of connected cells with little or no differentiation. You can have simple filaments, balls, sheets. And the colony can be attached and share cell walls, or they can actually be totally separate cells, just held together with connecting proteins. Here is an image of how more complex protists evolved from our more simplistic. And so with the protist, we have red, green, and brown algae, and then plants, fungi, and animals that all arose from our original protist, but they all arose independently with different single-celled ancestors, and we'll see this in our phylogenetic tree coming up. So we've looked at Volvox in the lab, or we're going to look at Volvox in the lab, and it is a multicellular green algae that evolved from a single-celled Chamaedomonas. Chamaedomonas was a unicellular algae, and then you can see its progression from 4 to 8 to 32. Here is a 16-celled. This is where they became balls. So rather than being sheets as they were in the beginning, they started forming these ball-like structures, spheroidal colonies with inversion, and then eventually into greater than 500 cells called the Volvox. And so here we can see the evolutionary progression from a simple unicellular up to a spherical 500-celled organism. And here is the phylogenetic tree. We have 4 supergroups. So we have the domain Eukarya, the kingdom Protist, and under that kingdom Protist, we have 4 supergroups that we're going to talk about. We have Excavata, making up the Diplomonatus, the Pelisas, and the Glenozonas. Our SAR clade, and this SAR stands for the Sturminophiles, Aviolates, and Rosarians. And so that is SAR. We have the dinoflagellates, ciliates, diatoms, a lot of our algae, golden and brown. Oomacetes are in here, the radiolarians, the forums. So all of those are under this SAR clade. The Arachoplastidia, or where we have the red algae and our Volvox and our chlorophytes. And this is where we derived our land plants. So land plants are not protists, but they came from this green algae protist lineage. And then the last supergroup, Uniconata. This is where we have our amoebozoans and our opsicanates. And the amoebozoans, slime molds, gymnoamoebas, entamoebas, all under here. And then with this other group under the Uniconatas where our fungi and animals came out. So these are not protists, but they derived from this arm of the protist tree. And so we will not delve any deeper into the phylogenetic tree other than these 4 supergroups. So protist, extremely diverse. Lots of different features, lots of different lineages, lots of different protists under that big umbrella. Most are unicellular, but we do have some multicellular, such as these kelp forests and some of our algae. And most of our single-cell protists can be extremely complex, as we just saw back with Euglena. Here's the Euglena, and we saw that it's actually a secondary endosymbiotic organism with 3 membranes, and is very metabolic, can do both photosynthetics and heterotrophs. So they can be very complex even as a single-celled individual. And here we can see some of the stentors that we looked at and we'll look at in lab. Our protists have varied array of cell surfaces. They have an extracellular matrix, and that extracellular matrix can be glass. So diatoms actually have a silica shell, which is a glass-like shell. They can form cysts, which means that they can survive very harsh environments, just as we saw with the endospores back with our prokaryotes. When they are in this cyst form, they can lie dormant for many years and can come out when habitat is favorable. Find this a lot in infectious protists, aids in that transmission. So here we see Giardia. So this is an intestinal parasite. In this stage, the trophozoite emerges, divides. You don't necessarily exhibit symptoms. That endospore, that cyst, can now form in the colon, can be expelled in the feces, and when the feces get into drinking water or something like that, or get on food, then are taken up by a new host, ingested, and it can start its life cycle again. I also have very many modes of transport. So we can have flagella, which is this one long tail. This is what we will see in Euglena. We have pseudopodia, pseudo meaning false, podia meaning feet. And so you have these false feet that you see in the amoebas. And our ciliates, you have cilia that are little short extensions that do wave-like motion to propel the ciliates through their environment. Have different modes of reproduction. We can have asexual reproduction, binary fission, just like our prokaryotes, where you get two identical equal-sized cells. You can have budding. This is kind of what we see in our Volvox. One daughter cell nucleus splits off from the parent cell, taking some cytoplasm with it, and forms a new individual. And you can have multiple fission, where a nucleus divides repeatedly, forming many daughter cells, and you get many offspring, but they're not going to be identical in size. They're usually going to be smaller. You can also have sexual reproduction. And so meiosis, this is what we see in humans to make gametes, but they can also undergo meiosis to make gametes. This allows for that genetic recombination in prophase 1 and random assortment of the chromosomes in that metaphase 1. And you can also have with sexual reproduction conjugation. So the same species come together, form a temporary union. And we saw this same term back with our protists, where they form the conjugation bridge and exchange genetic material so that they become genetically unique from the parent species. And some protists will move in between asexual and sexual life cycles. Lots of different ways in which they gain their food and their energy. So autotrophy, this is just like with the bacteria, photoautotrophy. Algae, Euglena, means that they use light to make carbohydrates from carbon dioxide and light. Chemoautotrophy is where they use a obtain it from a non-light source, so they have to obtain it from another organic individual. Heterotrophy, types of heterotrophy. Phagocytosis is where they engulf food particles, so here's phagocytosis, and break them down with usually something similar to a lysosome. Saprobes absorb nutrients from dead matter or organic waste, and so they can excrete enzymes outside the body and then absorb it. Or parasitic, where they actually live in the host, and as the host breaks down the food, they are able to absorb it from the host itself. And then mixotrophy means that you have both autotrophic and heterotrophic capabilities. Many of our prokaryotes are key producers in our food chains. And so our diatoms, dinoflagellates, multicellular algae, other protists, make about 30% of the world's photosynthesis. And so by 30% of the world's photosynthesis, there are big oxygen suppliers. Prokaryotes are about 20%, and plants are the other 50%. They can be in aquatic ecosystems, and the biomass of those photosynthetic protists can be available with nitrogen, phosphorus, or iron, so they can use other mechanisms to carry out their metabolic processes. They can be toxic when you get algae blooms. They can release a toxin, just like we talked back with the prokaryotes, that sometimes it's the metabolites that they make that are deadly, not the organism itself. And so when you get big algae blooms of the protists, they can create dead zones in many marine ecosystems. Get a lot at the mouth of the Mississippi River because of all the fertilizers that wash down the Mississippi and accumulate at the mouth, or the area where the Mississippi empties into the oceans. With all that nitrogen and phosphorus can cause the algae blooms. Some examples of our photosynthetic protists are diatoms. Those phytoplankton blooms, they are important for many of the ecological food chains within the ocean. And one way also that they can cause problems is when they bloom, the dead individuals fall to the ocean floor undecomposed, and that actually is a way for carbon sequestration. So the CO2 they remove from the atmosphere can be sequestered in their undecomposed bodies in the depths of the ocean. Symbiotic protists. So a lot of them form very symbiotic relationships. Our coral reefs are a symbiotic relationship with the dinoflagellates that live in the reefs, and they provide them with food and the color. But a lot of problems with the coral bleaching is because those rising ocean temperatures are killing the dinoflagellates that actually gave the coral reefs their color and their life. You also mentioned in a previous video we've watched are the termites, and some cockroaches actually have wood digesting protists in their guts that allow them to actually live off of the wood that they eat. You'll find these as in the group of parabasalids. But protists are parasitic too, causing many diseases. Phytophthora remora causes sudden oak death. P. infestans is related to the potato late blight. It's what caused the Irish potato famine back in the day. Also cause human disease. The trypanosomas cause African sleeping sickness. The plasmodium is what causes malaria. And then, as we've talked about before, algae blooms, red tide is where we get overgrowth of dinoflagellates because of all that phosphorus and nitrogen that can run off from the fertilizer supply.
Parasitism is a form of consumption in which one organism, the parasite, Parasitism is a form of consumption in which one organism, the parasite, consumes relatively small amounts of tissue or nutrients from another organism called the host. The Plasmodium life cycle involves the interaction of 3 species in a complex cycle that results in malaria in humans. There are 2 parasites in this scene. The mosquito acts as a parasite by taking blood from her human host. To ensure an even flow of blood, she first injects the site with saliva that contains an anticoagulant. The second parasite, called Plasmodium, flows in with the saliva. Plasmodium parasitizes the mosquito and the human, using each as a host for part of its life cycle. Plasmodium quickly travels through the circulatory system and reaches the liver, where it specifically targets liver cells. At this stage, the plasmodium cells are called sporozoites. A sporozoite invades a liver cell, where it divides many times to produce offspring that are known as merozoites. From the liver, these offspring enter the general circulation of the body. In the bloodstream, the merozoites continue the life cycle by infecting red blood cells. The invading merozoite lives off the hemoglobin and other nutrients in a red blood cell, allowing the parasite to divide and produce more merozoites. The merozoites destroy red blood cells and then find other red blood cells to infect. The destruction of red blood cells causes anemia in the host, a potentially dangerous consequence of being infected with the malaria parasite. Some of the merozoites in the bloodstream differentiate into male or female gametocytes. These cells no longer use the human as a host. Now they require a mosquito to continue their life cycle. When a mosquito bites an infected human, male and female gametocytes enter the mosquito as part of the blood meal. Once ingested, they enter the mosquito's gut. In the mosquito's gut, the haploid male gametocyte develops further, producing spindle-like male cells. These male cells fertilize haploid female cells, and diploid zygotes result. This diploid stage of the life cycle of Plasmodium migrates into the mosquito's gut to continue its life cycle. These cells divide by meiosis and then by mitosis to produce many haploid sporozoites. Sporozoites migrate to the salivary glands, where they're ready to be injected into another host the next time the mosquito bites a host. Now you've seen the entire Plasmodium life cycle and observed its development through two distinct hosts. In terms of evolution, what might you expect to happen over time in a human population exposed to the Plasmodium parasite? Humans are not defenseless against the Plasmodium parasite. We have evolved some immune responses that help combat infection. One example is shown here. Most cells of the body, including liver cells, display proteins called HLA on their cell surfaces. The specific types of HLA proteins depend on a person's genotype. Some people carry an allele called B53, which is particularly useful in combating Plasmodium. If a Plasmodium sporozoite infects a liver cell, the liver cell can use its HLA protein as a flag to call the immune system for help. During the infection, the liver cell puts small pieces of the parasite onto its surface. The HLA-B53 protein is unique in its ability to bind to a Plasmodium protein called CP26. Cells of the immune system called cytotoxic T cells can recognize the CP26 protein when it's displayed on the liver cell's surface. Cytotoxic T cells destroy the liver cell before the merozoites are released. By alerting T cells to the infection, the B53 allele of HLA is an effective adaptation against malaria. This adaptation is just one move in this coevolutionary arms race between the host and the parasite.
All right. So as we mentioned before, we have 4 supergroups of protists, and we'll get into this with lecture 3. Those supergroups are excavata, SAR, Arachiaplastidia, and uniconata. Plants arose from the Arachiaplastids, and fungi and animals arose from uniconata. Remember, the SAR stands for the Straminophiles, Aviolates, and Rosarians. See you in lecture 3.